are able to look
I've
at
analyzed, yeah, I've analyzed the entire genome sequence of this
Okay.
virus and compared it to the entire genome sequence of all the other coronaviruses that we have data for. And this weird element that doesn't belong there, I found that it actually did match a vector technology that was from the 1998 was the first time it was published in the Proceedings of the National Academy of Science. This vector technology is a mechanism by which molecular biologists insert new genes into viruses and bacteria. This vector technology is called a p-shuttle SN vector. Now, it's really unusual to find a vector technology sequence in a virus that's circulating in humans. And so naturally, one thing that we can say, I think for certain, is this particular virus has a laboratory origin. So we can
So what you're
rule out
saying is this technology is something scientists use to put in and insert things into the genetic coding of a virus to work with it. And what you're saying is that this is an actual product that scientists use that's now been found by you in what was said to be a wild virus. How would a technology end up into a wild strain virus? Is that put
That's exactly
clearly?
right.
Okay.
Exactly put clearly. And so it should not be in the wild animals anywhere. And in fact, if you then take that sequence and compare it to other proteins that it encodes, we find that it's actually a SARS protein that was put into a coronavirus for the purpose of making the vaccine work better. That's why this element, I believe, is in there. That's the science is showing us that element is in there to create a more reactogenic, a more immunogenic vaccine. So if that's the case, that doesn't also rule out a bioweapons origin where they may do the same thing for other reasons. So if we could talk for a minute about the patents, of course, there's patents for every virus on the planet that's been genetically modified. The world is swarming, laboratories are swarming with genetically modified viruses for research purposes, for the purpose of vaccines. Our country called for an end to the creation of these Frankenstein viruses that have more killing potential or are more contagious than their wild cousins in a very strongly stated article, specifically in 2015, that said, if we mess around with coronaviruses, we could get an accidental release. Now, the mechanism by which this particular virus made it into humans is unknown. Was it an escape from a bioweapons lab? We don't know. Was it somebody working in a laboratory that artificially, accidentally infected themselves? We don't know. Was it a vaccine that the Chinese government was experimenting with? And there's an interesting body of literature, Del, that shows that if you use a SARS vaccine in ferrets, in rats, or in monkeys, and then they get a secondary infection of a natural SARS virus, the elderly and the immunocompromised end up dying. It's like a two-hit problem. So what we may in fact be looking at is a hidden attempt by the Chinese to get a jump on this, the economics of vaccines, you know them full well. You cited them week after week.
I want to see that quote that just came up. Can I just read this quote really quickly that just came up? This is, these SARS-CoV vaccines all induced antibody and protection against infection with SARS-CoV. However, challenge of mice given any of the vaccines led to occurrence of Th2-type immunopathology, suggesting hypersensitivity to SARS-CoV components was induced. Caution in proceeding to application of a SARS-CoV vaccine in humans is indicated. So this thing basically, really simply put, this approach to vaccines isn't working yet. It's actually causing more problems than it's fixing, right? It's putting people at risk. Yeah.
It's not working no matter who develops the vaccine. And that's why the last time there was a SARS vaccine, remember SARS went away without a vaccine.
Yeah.
And it went away without the vaccine because the vaccine developers could not get grant funding because the mice and the ferrets and the rats were dying. Now, there's a very important distinction here. If this is a bioweapon, it's not only going to be, more able to induce disease, it's also going to be more easily transmittable. And we see that there is an increase, about twice as infectious as a regular SARS virus. And we don't know why. That's just been published, I think, about a week ago by Jonathan Reed in the UK. But let's imagine if it is a vaccine type that escaped a lab, or that's then everybody could get.